All right. Good morning, everyone. Good afternoon and good evening. Welcome to today's webinar. Thank you for joining us. Today's talk is entitled Phase 1 Trials in Oncology, Pros and Cons of Current Designs. My name is Kelsey Brown, and I am the Medical Writing Manager at TransPerfect Life Sciences in London, and it is my pleasure to be your moderator for today's session. So today's event will run for about 60 minutes, and that's going to include time at the very end for Q&A with our speakers. So please hold your questions or submit them throughout the course of the presentation through your questions panel. The session is designed to be interactive. Again, you can submit your questions in the chat box, or if you have any questions or need assistance with anything, just submit a question to me in your chat box directly. At this time, all participants are in listen-only mode. And just a quick note, the session is recorded and it's going to be made available to you on the IDDI website for viewing about 24 hours after the webinar is complete. This event is proudly hosted by the International Drug Development Institute, IDDI. IDDI is an expert center in biostatistical and integrated e-clinical services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI is headquartered near Brussels in Belgium with the U.S. Center of Operations in Raleigh, North Carolina. And now it is my immense pleasure to introduce you to today's speaker. That's Elizabeth Else-Court, Principal Statistician of Consulting Services at IDDI. Else graduated in bioengineering at KU Leuven in 1995, got her PhD in molecular genetics in 2003, and a master's degree in biostatistics at Ghent University in 2007. So after some years of experience in research and clinical biostatistics in diagnostic companies, Else then joined IDDI in 2011, where she conducted research related to biomarkers in Alzheimer's disease and validation of surrogate endpoints in advanced colorectal cancer. And then she then served as Director of Consulting Services from 2017 to 2019, and now focuses on consultancy activities. She's particularly interested in early phase trials using frequentist and Bayesian methods, as well as phase trials with biomarker-driven designs. And moreover, given her strong background in assay development, she is IDDI's expert in analytical and clinical validation of in vitro diagnostics. Else has published more extensively in Alzheimer's disease and biomarkers and oncology. And we're also joined by Dr. Everard Saad, Medical Director of IDDI. Dr. Saad has nearly 20 years of experience in medical oncology and clinical trial design. He graduated in medicine and trained in internal medicine in Sao Paulo and did his fellowship in medical oncology at the University of Texas MD Anderson Cancer Center in Houston. And then after practicing for several years, he shifted his professional career towards education and research in medical oncology and has a special interest now in clinical trial methodology, estimation of endpoints, and the development of novel therapies for cancer patients. So thank you everyone for joining. And now I'll pass this along to Else to start with our presentation.
Actually, it should be me, Kelsey. I will start.
Sure. There you go.
Thanks, Kelsey. So I hope everyone can see my slides. Hi, everyone. Thank you for joining this webinar. I will start by reviewing some concepts in dose escalation and some recent trends in phase 1 trials. So I'm going to spend the first 8 to 10 minutes on that. My part is really easy compared with what Else is going to do, because she is going to take up most of the presentation, and she'll talk about the taxonomy of current designs, their key statistical properties, and importantly, points to consider when selecting a phase 1 design. Now let's just review some definitions. So what we're calling here phase 1 trials is part of a family, let's say, of designs, possible designs. So historically, we have phase 1 We used to have already phase 1/2 trials. In some cases, when a new, for example, a combination was going to be tested with previously existing drugs, sometimes it was felt that it was sufficiently safe to combine the drugs and do a quick phase 1 and then move into phase 2. So we're not going to talk about that. And likewise, we're not going to talk, at least today, about what's become quite frequent these days, phase 1 trial with expansion cohorts. And the reason we won't talk about that will become clear along the way, because else we'll mention that toward the end. So what we are going to talk about is proper phase 1 trials, in the sense that their key feature is dose escalation. And it doesn't matter really if they are first in human. Often they are, but sometimes they're not. And it doesn't really matter about whether they're new agent combination or special populations. But it's true that many of the discussion and probably the examples are more applicable to new agents, but else we'll come back to the point of combination along the way. Now, phase 1 trials have two let's say, three big objectives, which are in larger font here. The first one is more quantitative and relatively technical and precise. So this is a lot of what else is going to talk about. And this objective is to determine the maximum tolerated dose, or MTD. Now, there are other objectives. The second one is a little bit more qualitative, so to speak, because defining the recommended phase 2 dose is not exactly rocket science, because it may take into account clinical considerations and other things that are related to the development program. But still, it's a key objective in phase 1. And characterizing the safety profile is somewhat of a vague statement, but it's a very important objective to have a good idea about what can be expected in the development of that particular drug or combination. Of course, we want to assess pharmacokinetics and pharmacodynamics whenever possible. And it used to be a smaller objective in phase 1 to assess activity, but this, as most of you know, is growing in importance and is driving a lot of the discussions in phase 1. But with very few exceptions, we'll leave this to another occasion, and our focus today is really on dose finding. Now, another word of caution regarding the MTD, because historically there are at least two possible definitions. Most of the times, phase 1 trials conducted in the US define the MTD as the highest dose level not causing DLT or dose limiting toxicity, however this is defined. So that the dose that did not cause toxicity in more than a certain percentage of patients. The percentage will vary. For example, in a 3+3 design, this is 33%. So that's not the point. But the point is that this was defined as a dose level not causing a certain level of toxicity, whereas in some studies conducted in Europe and in Japan, mostly in the past, the DLT was the dose level that would cause toxicity in more than X%, say 1/3 of patients. So here we are focusing mostly on the first definition. Where you put the equal sign is not so relevant, because as we'll see, this depends on the design. So the point here is not to emphasize where you put the equal sign. Now, all of this is based on the assumption that comes from the chemotherapy era, according to which there was a somewhat monotonic dose-response relationship, both for the anti-tumor effect as well as for the toxic effect, in the sense that you increase the dose, you increased the effect. Hopefully there was a good window here, in the sense that you would have sufficient activity before you had too much toxicity, but for both good effects and harmful effects, we would expect that there is this increasing monotonic relationship. Now, this gave rise historically to the famous 3+3 design, sometimes called modified Fibonacci design, according to which relatively simple rules for cohorts of 3 or 6 patients can be used to decide whether at a certain cohort you stay there, or you escalate, or you stop. So we don't need to go into the detail, but this is the regular, let's say the routine common setup of a 3+3 design, in which you will treat between 3 and 6 patients per dose level, depending on how things go. And this is the our starting point, let's say, for this discussion today. Now, the 3+3 has been criticized by many people in the past for various reasons, because it's relatively inflexible. It's one of the rule-based designs. It is not very statistically smart, let's say. It's relatively conservative in the sense that it tends to choose a lower doses as the MTD. It can be criticized because the dose escalation is relatively slow, because you will put at least 3 patients per dose level, compared, for example, with other designs. One of them is the continual reassessment method, or CRM, where you can go up much faster. And then there are imprecise estimates of the MTD, because in the end you will treat a few patients at the MTD. This graph shows the number of patients per dose level in a stepwise fashion. And when you see these signs, this means some patient has had a DLT. And so here you would have in the end very few patients treated close to what you would determine as the MTD, whereas with other designs you may be able to get more precision, because you will have more patients around the eventually recommended phase 2 dose. Now, in addition to those criticisms, in the era of targeted therapy, there have been other ones. Typically, the MTD is not reached with many targeted agents and, for example, monoclonal antibodies. And so there arose the concept of the maximum administered dose. And another potential problem, which is not specific to the 3+3, but is the fact that many of these novel drugs will have toxicity that appears a little bit later. And typically in phase 1, the concept of DLT was more restricted to the first cycle. But this is a more general criticism. Now, there are different potential dose-response relationships for targeted agents. So some people will conceptualize this relationship as on this panel here, whereas anti-tumor effect will continue to increase and the toxicity will follow a different rule. In some cases, there will be a kind of shoulder in the curve of efficacy, because there is saturation of a certain target, whereas the anti-tumor the anti-tumor effect will follow this saturation, whereas the toxicity may be more linear. It can be close to the to the efficacy in terms of the dose-response relationship, or hopefully it can be further away, the same way that I said about chemotherapy or cytotoxics. Now, we know that drug development is changing in many ways, and one of the most important is probably the fact that we're moving from a model of clear demarcation between phase 1, 2, and 3 into a more seamless let's say progression along the phases. And so efficacy, which was a consideration typically from phase 2 onward, with the exception of just preliminary evidence of activity, now efficacy is becoming more of a concern early on, and people are tending to incorporate the assessment of efficacy already in phase 1. And this is a very good review, not so new, but quite comprehensive in all the ways in which phase 1 trials in particular are changing, and one of these ways is related to the design chose for dose escalation. One would hope that there is less use of a 3+3 design and more use of designs that are model-based or model-assisted, as Els will illustrate to us. So now I'd like to hand it over to her, and I'll come back to the Q&A at the end. Els, are you on mute? Perhaps? No.
Can you hear me now?
Yes.
Yes. Yeah, I'm sorry. I have a problem with my headset that wasn't present just a while ago, but okay, if you can hear me like this, it's fine. Well, thank you, Everardo, for the introduction and for making it already clear that the 3+3 design is probably not the best design for dose finding, since it has many shortcomings. And in this table, I made a summary of potential features of the 3+3 design that novel designs could improve upon. And I will not go through all of them, but what I think is a very important one is the directions in which the designs can move. So the 3+3 design, it can only, once it has escalated the dose and de-escalated, it can only stop. It can no longer go up again and move across the dose levels. And this is what causes this conservatism and the underdosing of patients that is well known for the 3+3 design. And this is one of the features that I think the novel designs should improve upon by allowing the dose escalation to move in different directions throughout the trial. So a first set or a first class of new designs, well, I call it new because they are newer, relatively new compared to the 3+3 design. But the new set of designs are the model-based designs, and that are not as the 3+3, they are not based on ad hoc rules, but they are based on a parametric dose toxicity model. And here the parameter of this model will be updated after each cohort of patients has been treated. And the most used model-based design for the moment, I think, and one of the first was the continual reassessment method, or the CRM. So this model, yeah, I'm sorry, I have a cold and I might cough now and then. So this CRM model, it starts with a guess of a prior dose toxicity skeleton. So for each dose that you plan to take into the design, you need a guess for your toxicity rate that will be observed. And then you need to select a model that you will use to estimate the DLT probability. And here, what I show here, it's a simple one parameter model, also called the power model. And this model is then updated after each set or each cohort of patients is treated, the DLTs have been observed, and then you will have a new estimate of your DLT probability for the dose, for the different dose levels. And then you will assign patients in the next dose to this dose that corresponds to the estimated DLT rate closest to your target. And to make this a little bit more concrete, here we have a schematic illustration. So you have treated 3 patients, you update your model, this is the curve you obtain, and you see here that for the second dose level, you obtain a toxicity rate that is close to our target here in a dashed red line of 0.3. And so it means that the next patients, they will be treated at the second dose level. And then again, you take all the data that you have, you refit your model. And here we see we get the same type of curve. So it means that the next cohort of patients will be treated at the same level. But now no DLTs are observed, so there are no red balls here. And you see that the curve drops, meaning that now the third dose level is has an estimated DLT probability that is the closest to our target rate. And you see that then the next patients, they are treated at this third dose level. And you continue in this way until the maximum sample size is reached. Now, this design has some statistical desirable properties. It uses information of the different doses to select the next dose level. But what is, of course, of more importance, more important than the features of this design, is that when you look at its performance and compare it to the 3+3, it really outperforms the 3+3 as we would have hoped and expected. So it outperforms the 3+3 in the probability of correctly selecting the MTD and in the number of patients that are treated at or close to this MTD, and it does not cause more toxicity issues. There are no overdosing issues. Well, there were in the beginning, but there have been adaptations to the design. And so in terms of doing it does a better job, the CRM, in selecting the MTD. But there are also some disadvantages, and this is one of the reasons why the design is not, I think, so widely used, is that to get started, you need to elicitate this skeleton. And clinicians, they experience this as a kind of black box method. You observe DLTs, you put it in the model, and the model will tell you what the next dose level is you have to treat the patients on. So I think this is an important disadvantage. And also, well, simulations are needed to demonstrate how this model performs, especially to make sure that the model you are applying, the design you are applying, will not result in more toxicity. And it's a larger burden to get started. Another difficulty in implementing this design is that there are no pre-specified rules, as Everardo showed for the 3+3. It's really after treating a cohort of patients, there's data transfer, there's model update. And so if you don't have an in-house statistician, this can be challenging. So although this design has the good operational characteristics that I think are really the right ones for a dose finding trial, it did not convince clinicians to abandon the classical 3+3. But it is a benchmark, I think, for performance of other novel designs. So after those classes of algorithm or rule-based designs with the 3+3, the model-based designs, including this CRM that I just explained, there's a third class, big class of dose finding designs, and these are the model-assisted designs. And the objective of those model-assisted designs is to combine the best of both worlds. So to keep it simple, like the algorithm-based designs and the 3+3, so that you can pre-specify, you can tabulate the escalation and de-escalation rules in your protocol, and but at the same time use a model so that your decision-making is more efficient and that you have a superior performance in terms of selecting the right dose as the MTD. Because it's, I think here the important thing to remember about the model-assisted designs is that because you want the design to be easy and with pre-tabulated rules, it means that the modeling can only be done by dose and not like for the model-based designs as a CRM. You will not use all the data that you have been collected that far to come up with your next dose. It's just this one dose you are treating the patients at currently, and you will only use all this information to decide upon the next one. Here still, simulations are often deemed needed to show the characteristics of these designs, although maybe this, and I will come back to that, we see that there is a change in the need for performing many simulations, because people get more used, I think, to this type of designs. Now, these model-assisted designs are also called interval designs because in one way or another, they will define a target toxicity rate and an interval around it that's seen as desirable. And they will work with this interval. If you're outside of the interval, the dose will be adapted to obtain toxicity rates within the interval. All the model-assisted designs are the ones that I know, they are based on Bayesian arguments. And so it means that you treat a number of patients at a certain dose. And so here for the model-assisted designs, the model will only work at looking at the data of your current dose. You have the number of patients that experience a DLT at this dose, which is assumed to follow a binomial model. You define a prior beta distribution for this true DLT probability. So, you can also call this your prior belief of this true DLT probability. And combined with the data, this gives you a posterior distribution for the true toxicity rate, PJ. You will see this coming back in other slides. But because the designs, they are developed using uninformative priors, these posterior true toxicity rate is often just your observed toxicity rate. So if this sounds complex, I'll show you in the examples that to conduct a trial, you don't need to worry about, let's say, the Bayesian arguments that are behind each of these designs. Now, the first model-assisted design was a toxicity probability interval and a modified version thereof. I will not go into detail because we know by now that there's a high risk of overdosing with this design, and so it's no longer recommended. But it has, I think, the merits of trying to combine the simplicity with the model-based approach or with the sound decision making. I will go into detail for two model-assisted designs that are currently used and that are sound and safe to use. So I will give you some details about two designs and then talk about how to design, how to select one of the designs for your current study. So the first design I would like to discuss is the keyboard design. It was proposed to remedy the overdosing issue of the TPI. It's based on Bayesian arguments, and how it operates is that it will divide the probability line. So here you have a true toxicity rate ranging from 0 to 1 or from 0 to 100%, and it will divide this probability line in a series of equally width intervals or keys. So the keys, that's where the name of the design comes from. And then you define a target key that is the target dosing interval. So here, for instance, if you say that your target toxicity rate is 0.3, is 30%, then this would be your target key. And then after treating some patients and observing the DLTs, for instance, here, 3 patients are treated and 2 patients experience the DLT, you define your strongest key based on your posterior probability for this toxicity rate. And in this case, that is 66%. So you would end up with this key as this would be your strongest key or the interval with the highest probability of containing this true value of the toxicity rate. And then it's easy. You escalate if you see that your strongest key is to the left of your target key. You stay if your strongest key is the target key. And like shown here in this graph, if your strongest key is to the right of your target key, you would de-escalate. Now you also need to think about some additional rules to guarantee safety or to avoid exposing patients to a dose that you already are almost sure that it is overly toxic. And this is what is expressed here in this first bullet point. So a rule that's often used is that if this probability that your toxicity rate is above your target, if this probability exceeds a high number like 95%, then the dose is eliminated and also the higher doses. And then another additional rule that's often used in model-assisted designs is that you stop your algorithm if your next assigned dose has already a certain maximum number of patients, for instance, 9 patients, just to avoid that you spend or you include too many patients, whereas you already potentially might have found the right deal, the right MTD. So, but after all those say rules and Bayesian arguments, in the end, where this keyboard design ends up, it boils down to just an easy pre-tabulated set of escalation and de-escalation rules that you see here, and then the added elimination rule to make sure that there's no that patients are not exposed to toxic doses. And then another design, the last design I would like to discuss in detail is, I think, the most common model-assisted designs currently, and of which there are also many different adaptations, and then I will mention some adaptations later on. And it's easier, I think, than the keyboard design and more transparent. And what it does, it will divide here again this line of the DLT rate, so going from 0 to 100%. It will cut it in 3 using 2 thresholds, 2 pre-specified boundaries, a lambda E, an escalation, and a lambda D for de-escalation thresholds. And if you then observe a DLT at a certain dose that is within this stay interval, you stay. If you're lower, you escalate. If you are above, you de-escalate. So in terms of, I think, how this Bayesian design operates, it's very intuitive. Now, again, those boundaries for escalation and de-escalation, they are based on Bayesian arguments. And I won't go into detail, but what the design does, it minimizes the incorrect decision of dose escalation and de-escalation based on 3 different point hypotheses. And so for each of those 3 hypotheses, we need to define a parameter. And once we give this parameter, the Bayesian design will come up with estimates or with this derived boundaries for escalation and de-escalation. So with a typical choice, your thresholds would be like this. And so, for instance, if you observe 1 DLT out of 3, that's 33%, you would be in the stay region. If you observe 2 out of 3, 66, you are in the de-escalation region. And as explained for the keyboard design, there are additional rules that are added, and just to make sure that the doses that you already for which you already know that they are not safe or they have a too high toxicity probability, that they are excluded. And again, this results in this easy pre-specified decision rules. And here actually the table is exactly the same as the one that I showed for the keyboard design. So using different designs, using different rationales to come up with your decision rules, it still ends up in a same simple table. I think here what is also important to see is that these are the number of patients that are treated, and even if many of these model-assisted designs they are run with a cohort size of 3 patients, we do have rules for every number of patients that was treated at a certain dose level. So for instance, if you have a cohort of 3, but there's 1 patient that is no longer evaluable for DLTs, then you can just look at your decision rules for 2 patients, and you add 3 more in your next cohort. If it's the same dose, you end up with 5 patients, and you continue like this. So it's no longer, it's less rigid, and it's more flexible in terms of the number of patients you need for an evaluation. So to summarize, these model-assisted designs, they use a model behind the scene, but the inference is done at each dose level. So there's no borrowing of information across the dose levels during the actual dose escalation or de-escalation algorithm. There is borrowing of information across the dose levels when the actual MTD is selected at the end of the study. So when the study stops, all the data will be modeled using an isotonic regression. And here, the reasoning behind it is that you assume a monotonic increasing toxicity over the doses. So if for a certain dose you have, for a certain dose level, you have a toxicity level, a lower dose cannot have a higher toxicity probability. And that's what's taken into account when you establish then the MTD based on all the information that you gathered during the trial. So the trial continues until the sample size is exhausted, or if you have a maximum sample size at your current dose. And like I just said, so even if your cohort size is 3, there are rules for any number of evaluable patients. So again, those model-assisted designs, they seem to have the desirable statistical properties. It is simple because they can be expressed in simple pre-tabulated decision rules, but more importantly, it has been shown in many papers, in many simulations, that the model-assisted designs, they outperform the 3+3. And in most cases, they are as good as this model-based CRM, model-based designs like the CRM that I just discussed. And when I say superior in performance, I always mean in terms of probability of selecting the MTD and in number of patients treated at this MTD or close to it, and keeping it safe, so avoiding overdosing. So with those 3 different classes of designs, and I have just shown a few types, so within each of those classes you have many different designs. So when you need to design a study for your dose finding trial, you will have to compare operational characteristics of different designs. I think first you would go to literature, you would come up with some designs that would be suited for your agent that you think is what you want, and then you select some design types that you would like to compare, and also potentially some design parameters for the model-based and the model-assisted designs. For instance, if you have a Boeing design, question is, if we have a maximum sample size of 15, 18, 21 patients, well, does it improve our selection of the MTD if we increase this maximum sample size, or do we in fact have the same amount of information with 15 patients? So you will then need to simulate these designs under different plausible toxicity scenarios and compare the different designs in the different scenarios for important operating characteristics. So what are the sample sizes expected, the maximum, probability of overdosing, probability of correctly selecting this. And the regulatory agencies, they often expect this type of simulation report for model-based and model-assisted designs. Now here, an example of some toxicity scenarios that we have used for a certain a design when we were designing a dose finding trial. So you see here in different colors 8 different scenarios, and you see that most of them, they have a low toxicity and low expected toxicity rate at the first dose, and they go up slowly, more rapidly. But here there are also 2 scenarios that have already have the MTD at the first dose, or the first dose is here in the black scenario. It's already more than it's already overrun the MTD. And these are the scenarios that we add because regulatory agencies ask this, just to show that if your first doses are already overly toxic, that you pick the design that will spot this, and that you will halt the design without putting too many patients at risk. And then in terms of how we evaluate the simulations, so you see here the different graphs. Each plot is one operating characteristic. So here we have, for instance, average sample size. We have here the % correct MTD selection. And on the x-axis, you see the different scenarios. So you see that for here, for instance, for the first and second scenario, this 3+3 and Boeing design, they have the same average sample size, whereas for the other scenarios, the Boeing design has a larger sample size. You can also see that then here, when it comes to the percentage of correct MTD selection, that the Boeing is superior to the 3+3, so that for your increase in average sample size, you have a higher probability to select the right MTD. Now, maybe something I can point out here is that, well, if we say it is improved, yes, the blue line is above the red line, but still the percentage of selecting the correct MTD is around 50%. And I think this is, well, inevitably for this design with a limited number of patients per dose level, and that we will always end up, I think, with an imprecise estimate of the MTD. But there are some differences between designs, but we cannot expect miracles with designs that treat just a few patients at each dose. So some general findings when I compare the different designs, you can see these general findings are published, is that in terms of the model-based designs, the CRM will outperform the other model-based designs quite often. For CRM, skipping a dose is something that is no longer done, so because this would give you a higher risk of overdosing. And the Boeing and keyboard design, they perform similar and as good as the more complex, or more complex in terms of implementation, so this CRM design. It's interesting to see that the CRM or model-based design performs better when your proposed model is a good fit to the true dose toxicity, and when it's a bad fit. I think the model-assisted designs were shown to do even better from time to time. So this is about late onset. This is about the different classes of dose finding designs, but now I would like to talk about a few specific situations in which you would need to adapt your designs further. And the first specific situation is for an agent that potentially has some late onset toxicity, and that might give some logistical issues with the common phase 1 design, as we just discussed, because then you would have to suspend the accrual after each cohort and wait. So there have been some dedicated time to event designs have been published, and there is an adaptation time to event for CRM and for Boeing. And for instance, for Boeing, this is then how your decision table looks like. So you have your number of treated patients, your number of observed DLTs, but also the number of data that are still pending. And depending on those 3 elements, you will sometimes have already a decision to escalate, to de-escalate, or if too much data is still pending, there would be the rule to suspend accrual added. Another specific situation, and here I come back to what Everardo said, is that sometimes you would like to take efficacy also into the actual dose finding algorithm, because for targeted agents, we cannot just assume that more toxicity will mean more effect. So what is still often done is that common phase 1 designs only looking at toxicity are implemented, and first the upper safety limit is defined, and then to come up with the recommended dose for subsequent phases, you then informally look at efficacy and PK and other data that are available. Now, there are some dedicated designs that take both efficacy and toxicity into account. For instance, there's a Bayesian efficacy toxicity design. And so where here on top, I show the graph, the one-dimensional, say, decision rule for the Bayesian design, only looking at toxicity. This Bayesian ET has 2 dimensions, and it has an added dimension for efficacy. So if you would then, in terms of toxicity, for instance, be in the escalate zone, so you would be below this escalation boundary, but your efficacy is already above a certain predefined efficacy boundary, you would decide to stay because you already have sufficient efficacy. Now, another aspect of trials, of these dose-finding trials, is that sometimes drug developers want to incorporate historical data, which is not possible for the algorithm-based designs. For your model-based designs, this can be done in the specification of the skeleton. And for the model-assisted designs, we have another Bayesian. So here it's informative Bayesian. You can think about including this prior information by replacing your uninformative prior by this informative prior, and that for the doses for which you have information. So you would then end up with different decision rules for different doses because you have different you have different amounts and different prior information, but still you could use these predefined rules and still include historical data. The last specific trial I would like to discuss is when you have a combination of drugs for which you need to define the dose for each of the drugs. So it's not that you have one drug, for instance, a chemotherapy regimen that for which the dose regimen is already known, but now you have 2 drugs that you want to combine and for which you would like to find the MTD. And then, of course, there might be multiple MTDs. You could have, for instance, if you have for the both drugs you are at the 3rd dose level, you could have the same toxicity rate as having your one drug at the 2nd dose level and the other at the 4th level. And also, if you escalate and de-escalate, there are 2 directions in which you could make this shift. So one approach to this is the linearization approach. So where you select a sequence of combinations from the lowest dose to the highest dose for both your agents, and then you define the sequence and you apply a single agent design. So it means if you decide here to escalate, it's predefined that it will be in this direction and not in this direction. For Bayesian, there's also a drug combination design that's published. So it uses the typical Bayesian rules for escalation, de-escalation, but since 2 neighbors are possible, they will favor in this design the neighbor with the highest probability for the toxicity rate to fall within this desirable interval. And if there's an equal probability, for instance, if no patients are treated yet at those levels, then is chosen at random. And then to stop, I would like to come back to another point that Everardo made, is that often the phase 1 trials now include an expansion cohort in which efficacy or at least activity of the drug will be studied. And these expansion cohorts are, in fact, they are single-arm designs. In oncology, often they have as endpoint, they have the overall response rate. And these are small size trials. They compare the observed response rate to an historical control. And this is typically done with a design that has 2 stages, for instance, a Simon 2-stage, because at the time of you are conducting this trial, not a lot is known about the activity of the trial, and you would like to stop the trial after a 1st stage if there's really no sign of activity, because you don't want to expose more patients than strictly needed. And here, this is just an example of a typical Simon two-stage design for some alpha and beta levels and for some hypotheses on your overall response rate. You would come up with a sample size of 28. First stage would be 12 patients. And then based on the number of responses on these 12 patients, you would decide to stop for futility if there are not enough responses. And if you continue, you enroll more patients, and then you have your final analysis, and you will see if you want to take this drug any further. Now, if this two-stage design is added to phase 1 design, if you put those in the same model, then I think there are many ways of making this more compatible. For instance, if we have in this expansion cohort, we have 12 patients in this stage 1 to decide at interim to continue or not, we could make this compatible to the number of patients we have in on the selected dose in the phase 1, so that the end of the phase 1 is at the same time the interim for your stage 2, and you know already whether you need to enroll more patients or not. Because also the Simon two-stage design, it's just an example of a design, and there are many admissible designs with a different sample size at interim. So I think there are some synergies that we can find there. But I will leave this here. I will not say more about the phase 2 designs and expansion cohorts. We will do that in our next webinar that we will hold on the 2nd of December. And this will be dedicated to phase 2 designs. It will be a bit the same as today, really going over some different type of designs and discussing the pros and the cons. So then, just some fast conclusions. There are many different dose-finding designs available. I think the algorithm-based designs are seldom the best choice, and the model-assisted designs are often your best choice. The model-based designs can be the best choice if you have information, if you already have data on the dose toxicity relationship, and if you think it's logistically feasible. And to select your design type and fine-tune the parameters of your design, you can resort to literature, but you will always have to do your own simulations for the scenarios that are of interest to you. And then I think regardless of the design, we are looking in these dose finding trials at designs that have small sample sizes. So we can only expect that there is a high uncertainty on the selection of our MTD or the dose we select to take forward. And we have to live with these imprecise estimates of our MTD. So that is it from my side.
Thank you, Els. Thanks a lot for covering so much ground in so little time and with a cold.
Okay. I didn't talk too much.
All right. So I would encourage people to submit their questions. I already have one for you, very practical, Els. I guess it's more for you than for me because of your experience. Can you elaborate on your experience with regulatory acceptance of model-based and model-assisted designs?
Yes, I can. We have been interacting mainly with European agencies on model-assisted designs, and I see a shift. So when, like 5 years ago, when it was not a 3+3 design, then there would be all kinds of questions, and the simulations were not enough. We would need to do more. And now I think the latest interactions we presented the Boeing design to different European agencies, and there they just said, oh yes, we know the design, oh yes, we look at your parameters, it's good. So I think they are getting used to this type of designs, and they are much more comfortable with this type of design. So I think it's another reason why we should no longer do the 3+3 unless in very exceptional cases.
And can I add to that question, just piggyback on it? Are you talking about oncology specifically or more generally?
Yes, these were oncology studies.
Oncology.
Yes.
Okay. All right.
Yes.
Good.
And maybe,
Go ahead.
no, maybe what I wanted to say was that for model-based designs, I think the authorities will still require really extensive simulations because, yeah, there are more parameters to fine-tune and so forth, and they would be really looking into the performance of this particular design in specific situations.
Okay. Another very pragmatic question. Is there free software for these designs for computing sample
Well,
size and
yes. So I think the MD Anderson website will have many, has really great software for many of the designs I mentioned and for many others. If you would look into art packages that have been developed, or if you would look into the papers that I referenced, you will see that many statisticians that publish a method, a new design, that they have an art package already developed at that point in time. And we are at IDDI, we are working on some art packages that would combine what we think are the most interesting phase 1 designs, and that will also include some simulation features, but we are not there yet.
okay. Else, another question. How do you incorporate intrapatient dose escalation? Maybe I can just give my 2 cents first because that's something we didn't mention, right? And
No.
and that's something that I'll leave it up to you to talk about the, let's say, statistical feasibility of doing this. But from obviously from a patient perspective, in terms of clinical perspective, let's say, of trying to optimize treatment for patients, it's always a good idea. I don't think it's done very often, certainly not as much as one would like. And I know that I didn't show today, but there is some reviews that have been done in the past in which intrapatient dose escalation was a feature of more successful studies. But of course, this may be confounded by the treatment itself. So it's not that this is causal, but it's a good idea. That's what I wanted to say before you try to tackle this from a more statistical point of view.
I can see that it's a good idea, but you would need some, I think. So what I've been talking about now, it's only about decision rules after you have treated a cohort of patients. But if you would go into dose escalation within the patients, you would need to add some within patient rules, like when can you go forward? And I remember we have discussed this with one sponsor recently. And in the end, we found that it was, in practice, it was too complex, and we found another solution. So I'm not saying that it's not possible, but it would come at a cost of a larger complexity, and sometimes
Complexity.
it's more, yeah.
Right. Okay. Another question. In all these designs, are the dose levels defined by the sponsor?
Yes. So I think it's only the sponsor that will know the product well enough to come up with those estimates for the doses. Yes, and you would start with a dose for which you think that the toxicity rate or there's no toxicity, or if you are at a low toxicity rate, and you would go up until your presumed MTD or above. But yes, it's the sponsor that has the information to decide upon this.
Okay. We're getting close to the time, but there's I think there's time for one more question. Is there a role for randomization in phase 1? This is probably tailor-made for me because I like the topic.
I wanted to say, I think you are eager to answer that one.
Well, of course, always randomization, but then I have to ask the statistician if they can deal with it from defining what's the added value of having randomization in phase 1, for example, between the experimental agent that's being developed and some kind of standard control, or between 2 dose levels. I don't know.
I thought you would.
Well,
But
we like randomization because
yes,
there's always
as
more
early as
information to be
possible.
gathered, right? But
Yeah, yes, and I think, I mean, when we'll discuss like the phase 2 that is embedded within the phase 1 in the same study, sometimes you would like to have this randomization, especially for this phase 2, but then if the two are combined, you can as well start randomization earlier, and still, then you would include the patients not only for the dose finding, but also for the phase 2, meaning that there's an additional use of this randomized arm.
right. Okay, very good. We are very close to the hour. In fact, we're there. So I think for the two of us, that's it. I'll hand it back to Kelsey, and thanks everyone for joining us.
Yeah, thank you, Everard. Thank you so much, Alice, for joining and for presenting and doing the Q&A. We really appreciate your insights. And to everyone else, if we didn't get to your questions today, we'll be sure to follow up after this presentation. And if you do have any further questions afterwards, please direct them to info@iddi.com. That's info@iddi.com. And just one final note, you will be receiving a follow-up email from us with access to the recorded archive for this event. So you will have access to that recording. And thank you everyone for joining in today's webinar. We really appreciate it. And again, thank you both to our speakers very, very much. We hope you have a wonderful day, everyone in our audience. We appreciate your attendance and we hope to see you at the next one. Thank you, everyone.
Thanks, everyone. Bye-bye.
Bye.